Welcome back to Acing AP Biology. In this lesson, we will be introducing the topic of meiosis. Let's start by discussing the human genome. Human somatic cells have 46 chromosomes, each having distinct characteristics such as size, centromere location, and banding pattern. In a karyotype, which shows an individual's genetic makeup, chromosomes are matched in homologous pairs. Homologous chromosomes have the same size, centromere location, and banding pattern, indicating the same locations for genes. The locus of a gene is the specific location on a chromosome that contains a particular gene. So, homologous chromosomes have identical genetic loci. Human sex cells, or gametes, have 23 chromosomes, so they are known as haploid cells, because they only contain one set of chromosomes. Diploid cells, like somatic cells, contain two sets of chromosomes. Haploid cells are labeled with a single N, and diploid cells are labeled with two N. Gametes are the only cells in the human body that are not produced by mitosis, but are produced by meiosis. The processes of meiosis and fertilization exist in all sexually reproducing organisms. Life begins with a singular diploid zygote, forming after fertilization. Mitosis takes place, and over time, the zygote develops into a multicellular organism. Meiosis occurs during gamete production. Gametes are haploid, and no further cell division occurs in gametes before fertilization. Haploid gametes come together during fertilization to form a zygote. Fertilization restores the diploid number of chromosomes. Now let's go over an overview of meiosis. Meiosis produces gametes, which are haploid. The process of meiosis increases genetic variation. Prior to meiosis, chromosomes are replicated, and then two consecutive cell divisions occur, meiosis I and meiosis II. Meiosis results in 4 haploid daughter cells. In meiosis I, homologous chromosomes are separated, and this is known as reduction division. In meiosis II, sister chromatids are separated, exactly like in mitosis. Genetic variation increases in meiosis and fertilization in 3 different ways. Independent assortment occurs in metaphase I. Each homologous chromosome orients itself independently on the metaphase plate, so there is a 50-50 chance that the daughter cell receives the paternal chromosome and a 50-50 chance that it receives the maternal chromosome, creating genetic variation. Crossing over occurs during prophase I. It involves the reciprocal trade of genetic information between homologous chromosomes. Homologous chromosomes line up by corresponding loci, and genes are exchanged. The area where this crossing over takes place is called the chiasmata. And finally, random fertilization. The ovum carries 8 million possible chromosome combinations, and the sperm carries another 8 million. So, the zygote that results from fertilization has 64 trillion possible diploid combinations. That's it for this video. In the next lesson, we will go over the stages of meiosis. See you then. For more bio content, hit subscribe.